Hello, my name is Gabriela Imre, and I work at the Lifestyle Imaging Facility at Karolinska Institutet in Sweden. In this lecture, I will talk about lifestyle imaging, the challenges which come with it, and some recommendations on how to deal with them. What is lifestyle imaging? It's just as it sounds, imaging cells while they are alive, thus enabling visualization and quantification of dynamic cellular processes in real time. One of the most significant challenges in lifestyle imaging is to overcome photo damage. What is photo damage and where does it come from? When exciting a fluorophore, the fluorophore will transition from the ground state to an excited state, a state in which the fluorophore is highly susceptible for reacting with molecules in the surroundings, including oxygen. When oxygen interacts with an excited fluorophore, it will lead to an irreversible destruction of the fluorophore, and the destruction of the fluorophore is termed photobleaching. This will also give rise to reactive oxygen species. These reactive oxygen species will react with easily oxidizable cellular components such as proteins, nucleic acids, and membrane lipids, altering their structure and thus inducing damage to the cell. When shining light onto your sample, photo damage will occur not only in the focal plane, but also out of your focal plane, depending on what kind of imaging system you are using. In a wide-field system, the light is defocused, meaning everything is illuminated, so both below and above the cell we will have excitation and emission light. When using a single point confocal, the light is focused in one plane, but as you can see here, we do not only excite the focal plane, but also the planes below and above in the shape of a double cone, but to a much less extent than in wide field. This also means that fluorophores out of the focal plane will be excited too and bleached, thus inducing photo damage in the sample. All microscopes, with the exception of light sheet and multiphoton point confocal, are bleaching your sample also outside the plane of focus. Also, higher magnification objectives increases the illumination and will bleach more. So knowing how the light behaves when hitting your sample will help you understand where the photo damage will take place. Now that we know what photo bleaching and photo damage is, where it comes from and why, you will face a few experimental challenges when starting a lifestyle imaging experiment, and you should be aware of them already when designing your experiment. You will have to design your experiment and analysis pipeline in such a way that when imaging your sample, you are inflicting the minimum photo damage to your cells and still answer your scientific question. Cells are very sensitive to the light used to excite fluorophores, so it's very important to optimize the imaging conditions to minimize light toxicity as much as possible. This requires optimizing illumination power and exposure time, effective selection and collection of emitted photons. Of great importance is to provide the same environmental conditions, temperature, humidity, and pH conditions on the microscope stage as in the cell culture incubator. Another important aspect of lifestyle imaging experiments is to establish a reliable procedure to assess the health of the cells you are imaging during and after imaging. And even after you have checked all these factors, you should keep a critical mind when analyzing and interpreting your results. There will be circumstances when you will have to go back to square one, redesign your experiment and image analysis strategy, and make some compromises. So let's start with the experimental design. As all experiments, but live sample imaging in particular, require careful experimental design before starting the imaging. So what should you think about when designing your live cell imaging experiment? First of all, you have to have a very clear scientific question and define very clearly what you want to measure, meaning a feasible analysis strategy. Knowing how you want to analyze your data in order to get a reliable answer to your scientific question will help you decide on minimum resolution needed to reliably answer the scientific question, thus the choice of the objective you will use, choice of fluorophores that will work with the microscope you chosen, and ultimately sample preparation and imaging parameters. So how should you think when formulating the scientific question and analysis strategy? As I mentioned above, how you will analyze the images should be the core of your experimental design. Define very clearly what you want to measure and where. Let me give you an example. Let's say that you want to count the number of vesicles in your cells in an overnight live cell imaging experiment. At the beginning of your experiment, the dots seem not to be very crowded, and you can distinguish them individually and even count them. Thus, your imaging requirements will suffice using low NA objective, resulting in low resolution and contrast in the image. But you continue imaging, and by the end of your experiment, let's say 12 hours later, the field looks much more crowded with vesicles. You might think that you can still see individual vesicles, but if you increase enough the resolution and look at, for example, vesicle number 21 at time point 2, you can see that in reality it's not one vesicle, it's actually 7 vesicles. So now you need higher resolution and contrast in your image. So counting the number of vesicles at the end of your experiment means that you now need higher magnification, thus higher NA objective, and increased contrast. This will also mean higher illumination power on your cells, which means higher light toxicity. And even if you will be able to increase your magnification enough to see individual vesicles, it's still probably not possible to reliably count all vesicles anyways. So counting vesicles under these circumstances is not a very smart analysis strategy. Now, if instead you redefine the scientific question to match the limitation of your microscope, for example, if instead of counting individual vesicles, you reformulate the question and ask to know the percentage of the cell surface that is covered with vesicles. In this case, you do not need to identify each vesicle separately, so you will do just fine with having a lower resolution, meaning a low NA objective and ultimately less light toxicity, and on top of that a higher field of view, meaning more cells to analyze. So now let's see how we calculate the minimum resolution needed to reliably answer our scientific question. So how do we calculate resolution? As you already know, resolution is the smallest distance in the image that we can resolve. When the distance is not resolvable, for example, these two dots will look like one big dot. Resolution is limited by the objective numerical aperture, and you all know by now how to calculate the resolution delivered by the objective. Thus, knowing what is the minimum resolution you need to answer your scientific question, you will be able to decide on the objective you need to use. So let's see how we decide which objective we should use. Here we have two examples of live cells imaging. In the movie of the left, the scientific question required both high temporal resolution, frequent imaging, and high spatial resolution to identify focal adhesions. So the researcher used a 60x oil objective and barely fit two cells in the field of view. In the time-lapse in the right, The researcher did not need to specifically look at focal adhesions. She was only monitoring cell movement. Thus, in her imaging settings, she had low temporal and spatial resolution and used a 20x air objective. But this was enough to reliably answer her scientific question, and on top of that, could image many cells in one field of view that most to analyze. So remember, choose the minimum resolution needed. Choose your objective, magnification, numerical aperture, working distance, and ultimately the type of microscope so that you can reliably answer your scientific question. So now, let's see what should we pay attention to when choosing fluorophores. It's important to choose the best fluorophore for your experiment and your microscope, not just pick one randomly because it's available in the lab. This is true for all microscopy, but it becomes crucial for live cell imaging, because picking a suboptimal fluorophore will likely lead to longer light exposure and higher light toxicity than necessary, and thus unreliable results. How do we pick the best fluorophore? One chooses one type versus the other depending on the nature of the experiment. Fortunately, recent years have seen an explosion in the development of fluorescent labels and sensors for live cell imaging, thus allowing visualization of many cellular processes. Check the literature to find the latest fluorophore or fluorescent protein for your experimental needs. The excitation and emission spectra of the fluorophore or fluorophores you chosen must match the specification of the microscope you want to use, excitation and emission. And if you are imaging several fluorophores in the sample, you should not have any bleed through. On top of that, a key decision when it comes to picking the right fluorophore for your live experiment, it will also be determined by its optical properties. The fluorophore of your choice should have a high quantum efficiency so that it converts efficiently the absorbed light into emitted light, a large extinction coefficient to avoid high excitation powers, giving you a bright fluorophore, have a good photostability, and when it comes to fluorescent proteins, to be a monomer with a maturation time that should not slow down the folding of the target protein in the ER. It is not very likely that you will always have these characteristics at their best in one fluorophore, but knowing what they are, you will be able to compare several fluorescent molecules and pick the best for your particular experiment. And now we arrived at the last point in the design of the experiment, namely decision upon imaging strategy. What should we pay attention to when choosing imaging strategy? Ask yourself, can I skip some channels? Do I actually need to perform that steps? And how often should I image? And how long should my time-lapse be? For example, in this image, we used only one channel, and focal adhesions are labeled, and we clearly can see them. Although there is only one channel, it gives us a lot of additional information. For example, by pushing the brightness in the image a little bit, we easily see the cytoplasm, and this will help us segment the whole cell. We can also see a dark area where the nucleus is located, so we could also segment the nucleus if needed. All this information in one single image. So cut down any unnecessary sections, time points, or channels, and keep the minimum needed to answer your scientific question. By eliminating one element, you can gain light to use somewhere else. If you are using low illumination and your signal is very low, you will have a lot of noise in your image. Would you still answer your scientific question, for example, by using AI? Here we have an example where we use very low illumination power, and the objects of interest are barely visible due to the high noise in the image. but by applying a denoising algorithm, we can clearly identify the objects of interest. Just a summary of what we talked about so far. It was about designing the experiment. Always use reverse thinking. Start with the end. Refine the scientific question and analysis strategy. Define the minimum resolution needed to reliably answer the scientific question. After that, you will choose the objective which will deliver the required resolution. When you choose the fluorophore, you should choose fluorophore with efficient excitation and emission on the microscope you chose. If several fluorophores are present in your sample, it should not be any bleed-through between fluorophores. And only then, sample preparation, imaging, and image analysis. Now that we design our experiment and we have a feasible analysis strategy, it's time to work on how to minimize light toxicity when imaging our live sample. So how can we minimize light toxicity? First of all, by carefully choosing the fluorophores. Choose fluorophores with efficient excitation, with high photostability, brightness, and long excitation wavelengths. Avoid blue dyes, hexed if possible, for nuclear staining. Choose Drag5 or cDNA instead, with emission in far red. When imaging, you should choose a technique with low illumination. The choice of imaging modality greatly influences phototoxicity. If you can answer a scientific question with transmitted light, it's the best. The light power used for transmitted light imaging is much lower than for fluorescence. Choose an imaging system that gives the best signal-to-noise ratio to be able to minimize excitation power. Remember that cameras have a much higher quantum efficiency than detectors on single-point confocals. Additionally, the laser light in single-point confocals is concentrated in a tiny spot, thus the local illumination power at the spot is very high. If your sample is thick and you need 3D data, use a multi-point confocal or light sheet. Spinning these systems are much gentler for the sample than single-point confocals. In light sheet, the sample is only excited around the focal plane of the detection objective, thus not exposing the sample to light beyond the focal plane, and thus greatly reducing phototoxicity and photobleaching. Also, to avoid illuminating a double cone in Z, multiphoton illumination is a good option. Additionally, the long wavelength penetrates deeper in the tissue. Another option is to perform label-free imaging. How else can we minimize light toxicity? Your imaging medium can also induce phototoxicity and thus influence the well-being of your cells and the experimental results. Use cell culture medium with low flavins or vitamin B12 or phenol red. Vitamins, for example riboflavin and pyridoxal, have been shown to decrease photostability of enhanced green fluorescent protein, for example. When setting up the imaging parameters, use the lowest possible illumination and a short exposure time. Reduce the frame rate as much as possible. If necessary, close the field stop as far as possible to minimize the area in which the cells are exposed to excitation light. Choose long intervals between points, especially in long experiments. Also, remember that the Nyquist theorem works for time as well. Determine how long it takes for the function studied to show a significant change that can be reliably measured by image analysis. The optimal time interval is then half of that time, no more. And last but not least, do not forget to run control experiments for assessment of phototoxicity. Compare stain with unstained cell image in the same condition, and set up parameters for assessing the health of your cell, such as cell motility, division, mitochondrial shape, better than only assessing cell death. We will come back to this a little bit later. Now that you know how you want to image your sample, let's see if the microscope you chose to perform your lifestyle imaging would provide the same environmental conditions as your cell incubator. Because you will need to create and maintain the conditions that keep cells functioning and healthy during their entire time on the microscope. It is important to control the environment for optimal cell or tissue culture during imaging. Cells cannot tolerate large deviation from their optimal temperature, osmolarity, pH, and humidity. They will not die straight away, but they will first change their behavior, such as stop migrating or stop dividing. But keeping cells alive is not sufficient. We need to keep them healthy and make sure that the biological process assessed by the imaging experiment is not affected by the imaging itself. The temperature needs to be adapted to the sample. You will need different temperature if imaging mammalian cells or yeast or Drosophila. Allow a recovery time after transport between the cell culture incubator and microscope incubator. Make sure that you do not have any fluctuations in temperature during the time of your experiment, because changing in temperature will affect the well-being of the cells, such as protein expression, adhesion, cell proliferation, etc. Thus, you do not want to analyze any results which are due to temperature changes and not your treatment, for example. Check the pH of the medium at the start and at the end of your experiment. Air enriched to 5% CO2 is necessary for long-term imaging. Cells can also proliferate without CO2 if the medium is supplemented with HEPAs, but this environment is only viable for short-term imaging of a couple of hours. On top of that, for some cell lines, the addition of HEPAs needs to be gradual and done over a longer period of time before starting the imaging. A humidified atmosphere is very important so to minimize evaporation, as this leads to a concentration of the electrolytes in the medium and changes in pH. There is always more evaporation in a microscope incubator than in a cell culture incubator. Weigh the dish medium before and after the imaging session to estimate the evaporation. Evaporation or changes in osmolarity must be insignificant. If there is an evaporation problem, here are a few tricks that should help. First of all, use plenty of medium. Also, you can lay mineral oil on the sample as is done in fertility clinics. Mineral oil reduces evaporation a lot, and the good thing about it is that it is gas permeant, so oxygen can still reach the sample. If you use a multi-well plate, fill the outer wells with water. There is a well-known edge effect, where the cells in the outer wells behave differently from those further into the center of the plate. Also, humidity in ambient air can lead to condensation on all surfaces, especially on the lids of the cell culture vessel. If these surfaces are within the optical pathway, small water droplets will cause light scattering. Thus, must be avoided since it decreases the image quality when imaging transmitted light. Here we have an example of a fully heated enclosure around the microscope. This construction has the advantage of heating up not only the stage, but the whole microscope body, including the objectives, thus reducing focus drift and minimizing thermal cycling of optical and mechanical components. Sensitive equipment, like cameras, scanners, etc., are outside the incubator. In addition, the the system has a small chamber on the stage filled with humidified air with 5% CO2. This setup allows several days of imaging. There are also small stage incubators. The stage is heated up while the rest of the microscope is cold. Humidified air supplemented with CO2 is supplied to the chamber as in the large incubators. But a general problem with this design is the risk of condensation on the lid of your sample and the risk of focus drift, especially when using an oil objective. Thus, you should heat up your objective using an objective heater. These stage-top incubators are working well for short-term imaging experiments. Nowadays, you can also perform live imaging with small microscopes placed inside your cell culture incubator or using benchtop incubators with imaging capabilities. What else should we think about when it comes to the microscope of choice? Multipoint acquisition allows data collection from many fields of view, which is what we usually do in long-time lapse experiments or drug treatments. Thus, having a microscope equipped with a motorized stage will solve the problem. If you have an upright microscope, moving between different wells on chambers to image several conditions in one time-lapse will not be possible, due to the fact that the objective is above the stage, and moving between wells will not be possible. If performing live imaging with an upright microscope, you will be limited to a few positions within a larger carrier, for example a dish, and will also require a dipping lens objective. In contrast, when using an inverted microscope, since the objective is below stage, you can freely move between wells or chambers, and also have the freedom of choosing between different types of objectives. Focus drift is a notorious problem in time-lapse imaging. For short-term time-lapses, focus drift can be minimized by using good experimental practice. For example, by making sure that the specimen is securely seated on the stage and allowing a sufficient period of time for thermal equilibration before starting a time-lapse experiment. However, in experiments that last longer than a few minutes, this will likely not be enough. Thus, using an autofocus system is a must. You can use either a hardware autofocus that relies on detecting a reflection of a near-ear light beam from the interface between cover slip glass and imaging medium, and use drift of this reflection as a feedback for the motorized focus drive. Another option is software autofocus. This may be used on a transmitted light image, but not recommended in a fluorescence channel, because the focusing algorithms will need to acquire multiple additional images per time point to determine focus, thus resulting in rapid photobleaching. It is also recommended that the microscope used for imaging should be protected against undesirable vibrations by using, for example, anti-vibration microscope tables, especially for high-resolution imaging. When imaging, the choice of the sample carrier is of utmost importance. Nowadays, you can find imaging chambers for almost any kind of assays you might want to perform. Since the choice of microscope for live imaging is often an inverted microscope with the objective below the stage and thus in direct contact with your sample carrier, the bottom of your sample carrier should be made of glass with the specifications as number 1.5 or optical plastic also specified as number 1.5. This means a thickness of 170 micrometers. These choices will ensure high optical quality for imaging, but also low autofluorescence. In this graph, you can see the level of autofluorescence at different wavelengths in different materials. It is very clear that polystyrene which is a common material used for cell culture dishes and multi-well plates, have high autofluorescence in comparison with glass and special optical plastic. We did everything we could to set up our live cell imaging experiment with as little as possible light toxicity and on a microscope which fulfills the environmental conditions of an incubator. But we still need to have a reliable readout of the health of the cells throughout imaging, especially in longer experiments or when using short wavelength illumination. And the first step is to recognize photodamage. During imaging, cells suffer from photodamage long before they die. Unhealthy cells will show morphological distress, such as membrane blabbing, vacuoles, multiple nuclei, swollen mitochondria, or disrupted mitochondria, protein aggregates, as seen in this panel. Unfortunately, cell morphology is not a good readout, since some cells die instantaneously without changing shape. Here we have such an example, the cells in the red square. So when assessing the health of your cells during live imaging, monitoring dynamic processes such as cell division or cell migration is a good approach. Cells should be expected to perform these processes at rates as close as possible to the ones in the environment of the incubator. So here are some tips to what you should pay attention to avoid imaging unhealthy cells and draw wrong conclusions from your experiment. Compare the viability and doubling time of the cell line by comparing between culture incubator versus microscope incubator. Do the same by imaging with transmitted light versus fluorescence imaging. Compare the viability of cell image with 50% tougher and 50% gentler conditions than the imaging condition you need. For example, higher and lower laser power, double or half the time interval or length of the time lapse. Check the cells 24 hours after imaging to see if abnormal death rate or cell division occurred due to the imaging conditions. Check that the fusion proteins behave as published. Try several fusion locations, N-terminal, C-terminal, far from important domains, and compare cell division time, cell migration, occurrence of aggregates, etc. The stiffness of the environment greatly influences the cell behavior. Glass versus plastic. Not all cells are happy when growing on glass. Thus, they can change their morphology and moving pattern. Aside from the numerous problems associated with photodamage, as well as non-proper environmental control, you must also be alert to the possibility of microbial contamination during the course of your experiment. The most common contaminations are those that occur due to bacteria, yeast, mold, fungi, and probably the most common is mycoplasma, as visualized in the image on the right. The occurrence of many small dots outside the nucleus is a good indication of mycoplasma contamination, and it should be definitely confirmed by PCR. There are kits available to detect mycoplasma. There are solutions you can treat cells to get rid of mycoplasma. If that doesn't work, throw away the cell line and take up a new batch. After 1 or 2 passages, assess the cells for mycoplasma contamination. Mycoplasma infected cells do not die, but they behave differently from non-infected cells. And even after you have checked all the above-mentioned experimental conditions for live cell imaging, you should keep a critical mind when interpreting and analyzing your results. When using fluorescent proteins, you should pay attention to their behavior after fusion to your protein of interest. The fusion of a fluorescent protein to a native protein may change the protein's normal localization pattern or may lead to the formation of aggregates or oligomers. Most fluorophores dimerize weakly at high concentration, and so, when imaging, never image the brightest cell. Rather, go for cells expressing low levels of the fusion protein. Watch out for accumulation of fusion proteins in the endoplasmic reticulum. In Figure A, you can see an example of non-properly folded fusion protein which got stuck in the ER. Also, be aware that different fluorescent proteins may behave differently in certain cellular compartments. The GFP fusion protein localized to the ER network, and similarly, the MRFP fusion protein localized to the ER network as well, as seen in Figure C. But the MRFP tag protein also shows bright puncta, which are probably MRFP fusion molecules that did not fold properly and got stuck in the ER. So check the phenotype and localization of your fusion protein. And remember, only 70% of fusion proteins behave like the original protein. So critically observe your images. In summary, I will highlight the most important points you should pay attention to when starting a live cell imaging experiment. Have a very clear scientific question and image analysis strategy before starting imaging. Cells react sensitively to changes in their environment. Thus, it is crucial that the microscope environmental conditions are established to keep the cells healthy. The environment at the microscope must provide a precise and stable control of the temperature, humidity, pH, CO2 levels, and optionally oxygen levels. Keeping these parameters constantly in a physiological state will ensure optimal experimental conditions and produces reliable and reproducible results. Another important aspect when performing live cell imaging experiments is to minimize as much as possible light toxicity. Choose fluorophores with high photostability, brightness, and which work well with your filters at your microscope of choice. Use longer wavelengths, for example green or red, instead of UV or blue light excitation for fewer phototoxic effects. Choose a technique with low illumination and use the lowest possible illumination power and short exposure times. Reduce frame rate and choose long intervals between points. Also remember that the Nyquist sampling theorem works for time as well. Decrease the background fluorescence by using sample carriers with low autofluorescence and imaging medium specially revised for live cell imaging. Always remember, during imaging, cells suffer from photodamage long before they die, and some cells die instantaneously, almost without changing shape. Thus, only assessing cell health by monitoring changes in cell morphology or cell death is not the optimal approach. Rather, monitor dynamic processes such as cell migration and cell division. Cells should perform these processes at rates as close as possible as the ones in the cell incubator. Thank you for your attention.